This is the first notes on the DNA unit, and today it's all about DNA structure. So DNA actually stands for deoxyribonucleic acid. Now, I'm going to tell you what each of these pieces and parts of this word mean. If you find it helpful, you can underline the pieces, which will help you pronounce it. First of all, D means without. So D means without. Oxy, is short for oxygen. So whatever DNA is, it's missing an oxygen. So it's without an oxygen. Word ribo is short for the sugar ribose. They take the SE off to add the word to this big long word of DNA. So yes, our DNA has, every building block or monomer of DNA has one sugar in it. Then you already know nucleic acid. These are our macromolecules. And this is the macromolecule set that deals with directions. So deoxyribonucleic acid, DNA. And this is a nucleic acid that is made up of the sugar ribose, which is missing an oxygen. And I'll actually show you a picture of that later. The missing oxygen part doesn't mean too much to us now. So this is DNA. Now, we know from the cell unit that DNA is found in the cytoplasm of prokaryotes because they do not have a nucleus. And it is found in the nucleus of eukaryotes like us. All life on Earth is eukaryotic cell except bacteria. They're here. They're prokaryotes. We just said DNA is a nucleic acid molecule. That's the Na, nucleic acid. And it is arranged in pieces called chromosomes. In our eukaryotic cells, they're arranged in an X. So a chromosome, remember, has two chromatids made up of an X. Prokaryotic cells, they have one chromosome only, and it's made up of an O. One chromosome has 1,000 genes on it, roughly. So we have thousands of genes in our 46 chromosomes. One gene carries the directions to make one protein. Hair color, that's a protein. How curly or not curly your hair is, that's another protein, so that's another gene. Whether or not your hair is really thin or fine versus really thick or coarse, that's a different gene also. So there's all different kinds of genes, even just for hair color. Also skin color, how tall you're going to be, what color your eyes are, whether or not you're a fast runner or a slow runner, that's all coded for by your proteins, your genes. Now, here's a little video clip.
The instructions for building all living things, whether a fish, a bacterium, a tree, an insect, or a person, are found in a molecule called DNA. DNA is made up of 4 tiny building blocks. Each building block has a backbone and a chemical base that is represented by the letter A, C, G, or T. DNA molecules have 2 strands that are joined together through complementary base pairing. A pairs with T, and C pairs with G. These building blocks function together in larger units called genes. Each gene is a set of instructions for building a specific protein. A complete set of genes is called a genome. A genome is a set of instructions for building an entire organism. Every person's genome has the same genes arranged in the same order. But small differences in the sequence of the bases in our genes make each person unique.
Okay, this is chromosome number 1 for humans. It is the largest of all of our chromosomes. Every line going above this chromosome is the name of one gene. Every line below the chromosome is also one gene. Okay, we'll come back to this total a little bit later. It doesn't mean so much to you now. So let's look at cracking the DNA code. So moving along the first page, you know that a nucleotide is a subunit of a nucleic acid. That was in our macromolecule first unit. This is what a nucleotide or a building block of nucleic acids looks like. It is made up of one phosphate, which is always drawn as a circle. It's labeled here. One deoxyribose sugar. It is the sugar ribose, and it's missing an oxygen. Deoxyribose sugar. That's the blue house here. And then one nitrogen base, and nitrogen from the biochemistry unit capitalized with a capital N, and that's a base. These are the two, Dr. Watson and Dr. Crick. They discovered the molecule of DNA. They actually figured out what it looks like. The video that we just saw, they did all that. And they did that in the 1930s, and they won the Nobel Prize in the 1940s because when they discovered the structure of DNA, they didn't have a clue what it did. We didn't know what it was. We didn't know that it was a blueprint of life. So this picture is black and white because in the 1930s, they didn't have color film yet, okay? It's old. Crick passed some time ago. Watson just passed last year. He was well in his early 90s by then. So this is the structure of DNA. Kind of looks like a ladder. There are 4 nitrogen bases. A is abbreviation for adenine. C abbreviates for cysteine. T abbreviates for thymine. And G abbreviates for guanine. So just to pronounce saying these, adenine, say it, adenine. Cysteine, say it, cysteine. Thymine, like you're eating a chicken thigh, same kind of starting out the same way. So try to say thymine. And this one's always weird because the U is pronounced guanine. Guanine. And they are abbreviated by the first letter of each word. And here you can see them. And it's interesting, what letter or nitrogen base does A always pair with? Always pairs with T. T always pairs with A. What nitrogen base does G or guanine always pair with? Well, C, cytosine. And cytosine always pairs with guanine. That's the way it is. Okay, we don't know why. That's just the way whatever force you believe put all this stuff on planet Earth. That's what made it. Now, this picture you have in your notes, this is the deoxyribose, so I've labeled it for you. That's the orange thing. The phosphate is the little circles. And the backbone is this red box. So the video mentioned the backbone of DNA is made up of 2 repeating substances, phosphate, deoxyribose sugar, phosphate, deoxyribose sugar. Same thing over here, phosphate, deoxyribose sugar, phosphate, deoxyribose sugar. The bases or nitrogen bases, they are always in the middle. Again, that's just the way it is. These dotted lines are hydrogen bonds. We learned about them in our first unit. Do you guys remember, are hydrogen bonds the strongest of all bonds or the weakest of all bonds? Hopefully you remember they're the weakest of all bonds. And that's important because it is the bases, the order of these letters, these bases, is what the reason why you have the hair color you do, the eye color you do, why you're going to be only a certain height. It's the order of these nitrogen bases that does it. So it is important that this information be accessed quickly whenever it's needed. Now, you don't have to write this in the bottom of your notes because it's already printed for you. The code is the order of the nitrogen bases. So AGTC might give you lighter colored hair, whereas if you have GGGG, 4 G's in a row, that may give you darker hair. It's literally the order of those bases. Okay, now a codon is that's the name for one word in nucleic acid language. Now, you might think, well, that's just dumb. Why didn't they just call it a word? Well, there's a problem with the word. When we look at our English language, a word is varying lengths in every single language. You can have a one-letter word like I, a three-letter word like can, a two-letter word like go, and you can have a four-letter word, a five-letter word, a six-letter word. You can have words are a variety of lengths. Well, in DNA, it doesn't work that way. One word is always 3 nucleotides long. Never longer, never shorter, ever. Period. So what's a nucleotide again? Well, here's a nucleotide. 1 phosphate, 1 sugar, and 1 nitrogen base. You put 3 of these units together, you get 1 word in DNA. So all of this makes up 1 word in DNA. And you put 3 of them together. Okay, again, we don't know why, that's just the way it is. Okay, so our English language has words of a wide variety of lengths, whereas DNA language, it's only one. One way, 3 nucleotides make a codon. Now, let's practice our DNA pairing. So remember, your notes say from right here, A and T always pair together, G and C always pair together. So if you have one side of DNA reads ATCGGTATC, what does the other side read? So what letter bonds with A? Hopefully you said T. Well, if A bonds with T, what does T bond with? A. C. What does C bond with? It's not A or T. C bonds with G. What are those 2 G's going to bond with? C. What does the T bind with? A. A always binds with T. T always binds with A. And the last one, the C binds with a G. Okay, so this is actually a question on your test, your next test. We're going to give you a string of DNA nitrogen bases, and we're going to tell you what does it pair with? What's the complementary or opposite side? So let's practice this again. So if DNA reads this, what does the complementary side read? Complementary means opposite, and I'll have to write that in a minute, so it won't let me cheat. So, gonna go faster this time. What nitrogen base always bonds with C? What always binds with G? What binds with C again? What binds with A? What binds with G? What binds with C? And I think you guys get it by now. And I can't do anything about the periods, guys, sorry. All right, now complementary, means opposite. So when they ask for the complementary side, if they give you a C, you're always going to write a G. They give you an A, you're always going to write a T. They give you a T, you're going to write A's. Okay, so that's the way it works. All right, nitrogen bases. The way it works, these are your nitrogen bases. This is a big base. Adenine is a large base. Thymine, its mate or pair, is a short base. Guanine, G, is a long base. Cytosine, C, is a short base. And this is interesting. The way it works, the bases are like puzzle pieces that fit together. You always have to have a long base and a small base. Long and short, bonding together. That's the only thing that fits the space across the DNA ladder. So if the backbone are your sugar and phosphates, then like the letters, the nitrogen bases are the rungs. Always have to have a short base bonded with a long base. That's just the way it was made. All right, now, DNA replication. We just took our test on this. DNA replication, replication means to copy. Okay, and here, complementary means opposite. So the way DNA works is this is our DNA. We're going to call this our original strand of DNA, the clear strand. It has no color. The first step that happens is the DNA unwinds, and then all those hydrogen bonds, all the hydrogen bonds break. So all these hydrogen bonds right here, as the DNA untwirls around itself, they just break apart because remember, they're really, really weak. And then in the next step, new nucleic acids, new nucleotide units are put in opposite of the old. So that would be your darker strand. And I have a video of this because the video makes it a lot easier to see.
DNA replication is defined as the process by which an organism's original DNA is used as a template for the production of a new complementary DNA strand. An enzyme called helicase unwinds the original DNA's double helix, creating a replication fork. Next, an enzyme named DNA polymerase III works down the leading strand and up the lagging strand of the replication fork, synthesizing two new strands of DNA by taking free nucleotides and pairing them with the complementary bases on the original DNA template. The process of DNA replication is described as semi-conservative because the two copies of DNA produced each contain one strand of the original DNA and one entirely new DNA strand.
Okay, so in this video, an enzyme actually breaks the helix apart, and it breaks those hydrogen bonds and exposes the bases. Okay, then new nucleotides are brought in, and a different enzyme bonds them all together to create a new strand. Now, they called this semi-conservative. Here's why. This is the original strand of DNA. This strand of DNA has already been used in the cell, so the cell knows it works. So it makes a new strand from the old strand that is already a functioning strand. So that's why it's called semi-conservative, and that just means it just makes less mistakes. Okay, now, last thing, when in the cell cycle is DNA replicated? This is what we just had our test on. So this is during G1 phase. Do you remember which part of interphase does the DNA replicate? Is it S or G2? It is going to be S phase. And notice you have one chromatid here. Now it has been copied. You have two chromatids of the red, which may be mamas, two chromatids now of the blue, which are daddies. And then in mitosis, they split. So you have these two cells are identical to this cell. Okay, so that is DNA and notes. So you will have a formative assessment to do next.